from Singapore. Hello, thank you very much for coming. And I do know it's 8 o'clock on a Thursday, so I'll try to keep it as light as possible, but there are a bit of biological terms that I'll be chatting about today. So I'm just going to share my screen to my slides and let me know if you guys can see this okay. Yep. Okay, so as Jiashan mentioned, my name is Kai. I'm a third-year systematic ichthyology PhD student at the University of Sydney, and I'm also a research fellow at the Australian Museum. So what I do basically is I kind of work on taxonomy and systematics. So that's the science of describing and naming new species of fishes, and also working out broader evolutionary relationships between said species and other species that they're related to across temporal space and time. Yeah, and I'm in my final year of my PhD in the molecular ecology, evolution, and phylogenetics lab at the University of Sydney. And I previously did my honors in the same lab as well. Hold on, let me just get rid of this. There we go. Yeah. Yep, so aside from taxonomy and systematics, I'm also quite interested in general processes such as historical biogeography, phylogenomics, population genetics, and a bit of natural history. And these are all really exciting fields because on their own, they're quite distinct entities, but together they fit quite nicely together in the broader framework of systematics. Yeah, so initially I was going to present on a single topic, but given the duration of this talk, I figured that I maybe give a little bit more background to what I've been doing across the last 3.5 years of my PhD, and then also try to integrate this into a continuous narrative. So I'll split this talk into 2 different parts. And the first part, I'll talk about the new discoveries, and that's pretty self-explanatory here. I'll detail new things that are hidden in plain sight, ranging from new species discoveries and other discoveries that are broader, such as ecosystem processes, and also distributional records. And in the second part of the talk, I will be talking about systematics. And here I will present some case studies where we look at relationships of selected taxonomic groups, where I've integrated biogeography, morphological data, and also phylogenomic techniques in trying to tease apart the evolutionary history of these fishes. So you could probably already guess that most of the data-driven part of this talk will be in the second half, which is good, I guess, because it gives me a bit of time to slowly ease into the drier, more boring part of this talk. Okay, yep. Okay, so here's just a brief breakdown of the contents for today's talk. Yep, so for the first part, like I said, I'll talk about mesophotic coral ecosystems, what it is, why it's important, why we should care about it, as well as the new species that are living there and the discoveries that we've made over the years, and finally, hybridization on coral reefs. And for the second part of this talk, I'll give a brief overview of marine biogeography, followed by case studies on the systematics of 2 labrid genera. So I work on labrids quite a bit. You guys might know them as wrasses. And I'll give a case study on 2 different genera that I've been working on for the last couple of years. And I will also very briefly touch on some biogeographical patterns known as antitropicality. So I know that most of you here are probably already familiar with this, but just as a refresher, here's a map, a very, very basic schematic map of all the major oceanic bodies around the world. And for the interest of this talk, I will only be referring to the Indian and the Pacific Oceans, which is colored here in orange and purple. And that's only because I don't really work on Atlantic fauna. So I will just leave that out for today's talk. Yep. And as you know, about 70% of the Earth's surface is covered in water, and nearly all of that is found in our oceans and seas. Understandably, these systems are some of the least explored, particularly in the context of coral reefs. So hundreds of new species are continuously being discovered, and rediscoveries are also just as common as are new insights into general phenomena. And a little bit more background about mesophotic coral reefs. These are reefs that sit between 30 to 150 meters usually. So at this depth, it's a little bit too deep to explore with conventional methods of scuba diving, but it's also too shallow for most submersibles. So it sits at this little band around tropical coral reefs, which we sometimes call the twilight zone. And inhabitants here, they're extremely complex. We know very little about them. And also the coral reef ecosystem itself is very complex. with very intricate matrices and coral colonies and structures, which makes collecting really difficult. So when you combine all of these factors together, it's really no surprise that mesophotic coral ecosystems hold vast amount of discoveries that are just waiting for us to tap into. And traditionally, these deeper reefs were thought of as a refuge for the shallow water reefs above them, where they protect shallow water species from natural and also anthropogenic disasters, thereby promising the survival of shallow reef inhabitants. However, we know today that that's not the case because mesophotic coral ecosystems are actually ecologically quite distinct from the shallow water counterparts, with faunal assemblages, including fish and coral, that differ quite remarkably from species that you find living in 5 to 10 meters, for example. So this is a figure from Luis Rocha's group from a paper published in Science in 2018. And here you can see some examples of shallow water inhabitants in panel A compared to some deep water mesophotic species in panel B. So mesophotic reefs can be broken down into 3 zones, the upper, middle, and lower mesophotic. And in general, the deeper you go, the more dissimilarities you observe from the shallow water reefs. And it's important to note that these deep reefs are also under the same stresses that affect shallow water reefs and are in need of as much protection as their shallow water counterparts. I should also add here, though, that I am not qualified to do mesophotic diving. To do so would require very rigorous training and specialist equipment, both of which I don't have any background in. I, however, do collaborate with many prominent veteran and also emerging mesophotic biologists, which I'm very thankful for. And so they will feature quite heavily in my talk today. So mesophotic coral ecosystems are therefore a treasure trove of new species. And this panel here are just all the new species that I have described between the years of 2016 to 2018. So within a 5-year period. And of the 22 species here, 14 of them, so just over 60%, are found only in mesophotic coral ecosystems. And those are the ones that are labeled with the blue circle beside their names. And the orange dots are just the ones that for which I'm the lead author for that paper. So I won't go into too much detail here for each species, but it's nice to note that for most of these, they come from the families Labridae, Serranidae, and Pomacentridae, which also happen to be amongst the largest families of coral reef fishes. There is one species of goby here, and gobies are the largest and most abundant family of fishes in all aquatic habitats, but I don't work on gobies very much, so it's a bit disproportionate here. But yeah, the take-home message is that there are tons of new species living in these habitats, and a large portion of them are from really big families that we think we know a lot about, but we're actually just scratching the surface. And while new species discoveries are interesting, rediscovering old ones are sometimes just as good. So this was a paper that I collaborated with last year with Patrick Plantard and Brian Green on mesophotic fishes of the Reunion Island. Patrick is a mesophotic diver, so he's not a biologist, but he is a wonderful a wonderful photographer, and he's very good with natural history and a really good naturalist. And he's one of the few people doing mesophotic explorations in the Indian Ocean. And for those of you who are not aware, the Reunion Island is located in the Mascarene Archipelago in the Western Indian Ocean. And so if you're comparing the Indian Ocean to the Pacific Ocean, there is a lot less research being done on mesophotic ecosystems there than anywhere else in the world. So we published a short communications where we reported the presence of this really rare angelfish, which is the yellow one on the right panel, which is known as Apolomictes guzii, and it's only known from the type series described in 1978. And then afterwards with a single underwater photograph taken in 2003. So basically 25 years and then 17 years after the initial description. And this was the first time we're actually seeing really good photos of this fish. And because Patrick is a really good underwater photographer, as well as a really good underwater diver, yeah, he got a whole bunch of really great data about its biology, about its habitat, and also really good high quality photos. And he basically sent me an email saying, hey, I photographed this really cool yellow angelfish from the Reunion Islands, and I don't really know what it is. Do you have any idea? And I knew instantly what it was. So we got together with Brian Green and we wrote a little paper about this. And along the way, we also reported 2 new species from the Indian Ocean as being, new records for the area, which are previously not found there. So a really good interesting study, a natural history study with very little, with very little that we went off on, but with big implications on the distributional record of really rare species from really poorly explored areas. And I'm also really big on science communication. So throughout the slide, I'll be talking about projects that I have had the luxury of working on that were born out of collaborations through Twitter and Facebook and stuff like that. So this is just one of several. In the same year, I was also really lucky to be part of the Schmitz Institute visioning the Coral Sea Marine Parks project. And I think some of you on here might have seen that on Twitter, as well as ABC News in Australia. And this was a project that was in collaboration with several academics from James Cook University, Sydney University, Queensland Museum, and the Australian Museum as well. And for reference, the Coral Sea is in the northeastern part of Australia, just where the Great Barrier Reef is located. And this expedition was a little bit unusual and different from the ones that I've participated in that there was no diving involved. So instead, a remote operated submarine that was tethered to the research vessel, equipped with a 4K video submarine robot, basically, was used to map the seafloor, in the process picking up footage and photographs of fish, coral, and invertebrate life that were not really often seen. And as the principal investigator of fishes, this was a really exciting project. And throughout our expedition, not only did we identify putative new species, but also new records for Australia and the greater Pacific region. And this project is actually currently still ongoing, and the Schmitz Institute of Oceanography is still planning dives throughout the year and incoming years. And we're currently working on compiling a checklist of fishes observed from this trip and others. And like the previous study, this one was also born through social media. The expedition started out focusing on geology and deep reef chasms, mapping on the seafloor and identifying volcanic rocks. But the crew were picking up a lot of footage of fish, coral, crabs, the sort of thing. And so they kind of took to Twitter to crowdsource scientists who might possibly know what these are. And along the way, picked up a few scientists who they reached out to and said, hey, would you like to be part of this expedition? It's COVID, it's remote, you don't have to be shipboard, just call into the live stream and narrate what you see as we go along. So I said yes to this, and I've met a bunch of really, really talented and amazing scientists who are, wonderful to work with. So this has been a really invaluable insight into the reefs of Australia, and also just being able to work with people in a COVID-driven world that is, unfamiliar to myself and everyone here, I'm assuming. But not all new discoveries are confined to the deep. And this was another really fun collaboration that was born out of a Twitter message, if you can believe. So I got a message from Victor Brun via Twitter asking for a fish ID. And I was actually stumped when he sent me this image of this alarmingly red tiger stripe fish, which he found in an artisanal fishery landing site in Palawan, Philippines. And so this fish clearly belonged to the genus Labridae, which is kind of dotty back from the family Pseudochromidae. But I wasn't very sure what species it was. So with the help of Kent Sorgan, who's actually here tonight, hello Kent, thank you very much for coming. We managed to collect a bunch of specimens and send it to one of my taxonomy mentors, Dr. Anthony Gill, who confirmed that this was Labridae atrofasciatus, a really, really rare species that was described only from a single specimen in 1933. So it's been almost 100 years since another specimen has been found. And so we wrote up a simple paper describing new life coloration details, new distributional records. And we published it in the Raffles Bulletin of Zoology here in Singapore with Kent, Victor, and Anthony Gill. So that was a really interesting study. So previously I said that the fish was only known from a single specimen in 1933. A photograph of a putative female was published in 2003, but it wasn't collected. And beyond that, there hasn't been much in the literature concerning this fish. So with our specimens, we were actually able to kind of deduce the sex of the specimens and also corroborate what we know with what has been published in the form of the single photograph. We also took DNA specimens, we barcoded them, and the barcodes are now up online for other scientists and researchers to access. So yeah, this study was obviously interesting for several reasons. The first was that the discovery of the males which we detail with color photographs in the paper that has never been seen before. And also the other thing was the interesting biogeography. So Labridae atrofasciatus is found in Palawan. That's a region in the Philippines that is starting to look like a possible localized hub of endemism. And this general area is also home to 3 other endemic dottybags and a handful of other coral reef fishes. But most importantly, it's remarkable that such a big and charismatic fish that's living in shallow waters can evade detection up to now. But then again, also hold really important implications into the management of artisanal fisheries and also the protection of potentially endemic taxa found in smaller satellite islands of the Philippines. And so I'll end off the first part of my talk with hybridization, which is a very well-known phenomenon globally across most of the tree of life, but a phenomenon with a disproportionate gap in the literature. So comparatively little is known about hybridization in marine ecosystems compared to terrestrial ecosystems. And as you can imagine, in marine ecosystems where physical barriers of isolation are a bit more attenuated, you would expect there to be more examples of hybrids between sympatric species. And what are sympatric species? Sympatric species are just species that live in overlapping distributions. Yet the bulk of marine hybrid studies have focused on species that are parapatric, which means that they live adjacent to each other and don't really share distributional overlap. And relatively little is known concerning sympatric hybridization. So here's just a little bit more about sympatric versus parapatric distribution. Species with parapatric distributions are those that occupy distinct geographical range, but with their ranges adjacent to each other. And at the interface of these 2 regions, both species can sometimes mix and come into contact. And it is in such situations that hybrids in coral reef fishes are most observed, leading to these contact points being referred to as hybrid zones or suture zones, or even hybrid hotspots. So in panel A, I have an example here where the orange species and the blue species are found in separate ocean basins, but with both coming into contact at the boundary, where I've colored purple in Indonesia. And sympatry is a much simpler term, and it refers to species which overlap either completely or for most of their ranges. And both of these models are, of course, in stark contrast to a third term known as allopatry, where there is absolutely no contact at all between species. So we wanted to stress that the term sympatry can sometimes also refer to species that overlap in distribution, but are otherwise separated by ecological niche. So species that are found, for example, in the same region, but don't ever mix because they live, for example, in shallow water reefs versus mesophotic reefs, which is sometimes known as microallopatry. But this term is slightly problematic because it conflates a number of geographical and ecological concepts. So the defining meaning for allopatry is the separation of species by discrete extrinsic barrier to dispersal and not by an organism's intrinsic biology or ecological preference. So in this context, gene flow is restricted by a biological barrier rather than a mechanical barrier. And for this particular study, we define a hybrid zone to be a purely biogeographical scenario or context. Okay, I'm seeing a few questions here, but I will address them at the end of the talk because so thank you very much for your questions, but I will address them at the end of the talk. So yeah, the main question we wanted to ask here is how prevalent is sympatric hybridization on coral reefs, and to what extent can hybrids occur given the extent of genetic divergence between parents? And amongst coral reef fishes, a considerable amount of research has been directed at the butterfly fishes. Why? Because they are conspicuous on reefs, they're very colorful, and as such, it makes identifying hybrids very easy. So on the screen here, I have an example of 2 different butterfly fish hybrids, 2 different butterfly fish species, Chaetodon ophibium and Chaetodon auriga. And you can see on the right, the hybrid of both species. And it has traits that identify with one or both parents. And we refer to this as an intermediate pattern where you can identify, or really you can kind of roughly guess who the parents are based on what phenotype it's exhibiting. And so up to 39% of butterfly fish species are known to form hybrids, of which 90% occur in parapatric species. So here we have this big knowledge gap then of like what's happening with other fishes outside of the butterfly fish family and what's happening with them in sympatric regions of overlap. So we used another model, coral reef group, as a proxy to test this. And we used the marine angelfishes. Like the butterfly fishes, they are also very conspicuous, which makes identifying hybrids pretty easy. So you can see here, I've given you 4 examples of parental species and their hybrid crosses. And the hybrids all show really identifiable intermediate phenotypes that identify with both parents. So it's kind of like a 50-50 mix between what's mating to produce them. So in short, we surveyed the literature as well as photographs taken by divers, aquarists, other scientists, and we looked for hybrids that shared coloration patterns that aligned to one or both parents. And the angelfishes, aside from being colorful, also fulfill many other criteria that allowed us to test in this study. Number one, it is a very specious and very conspicuous group with many species from many genera. There are numerous examples of sister species that occur in parapetry, allopatry, and sympatry, which makes this group an ideal candidate to explore hybridization. So the angelfishes, not only are they very widespread, not only are they very specious, they conform to every single known biogeographical model. And lastly, there are numerous species that are endemic to hybrid zones, which allow us to give the butterfly fishes a fair comparative in terms of parapatric examples of hybridization between sister species. So what we did was, for every putative hybrid that we identified, we then looked at the genetic distance between each contributing parent. And then we mapped this out, and we found that hybrids occurred between parents with as little as 0% genetic difference. In other words, parents that are completely introgressed, and I'll talk about this term in a little bit, to parents that have as much as 12% genetic distance, which is very, very high. So in most terrestrial ecosystems, hybrids usually fall between parents that are between 2 to 6% divergent in their genetic material. So 12% is alarmingly high. And we also look at the geographical distribution for each contributing parent and then determine if they had sympatric or parapatric ranges. Now, I want to stress here that when we say genetic distance and genetic percentage, we're talking about mitochondrial CO1. So we're only looking at a specific barcode in the mitochondrial DNA and not the entire genome, which is the standard for most studies, unfortunately, just because entire genome sequencing for all these species would cost a huge amount of money, and it's just not practical at the moment. So we wanted to make sure that our method of visual identification of putative hybrids were robust. Otherwise, this entire study would be very speculative. And to do this, we picked a putative hybrid that was very clearly a 50-50 mix between its contributing parents. So in this case, we picked a hybrid between Paracentropyge multifasciata, which is the angelfish here with the white and black stripes, and Paracentropyge venusta, which is the one with the purple back and the yellow band. And to confirm if this individual here with the squiggly pattern is truly a hybrid between both species, and to see if our methods of visual identifications were reliable, we did a mitonuclear analysis using mitochondrial CO1 and nuclear markers such as RAG2, nuclear R7, and TMO4C4. And unsurprisingly, the hybrid had nested CO1 sequences that align with Paracentropyge multifasciata, and it shared nuclear haplotypes with both species. So what this basically means is that the squiggly specimen we saw here is indeed a hybrid between both species, with the stripy one being the mom and the blue back one being the dad, because we know that mitochondrial DNA can only be maternally inherited unilaterally from the maternal side. And because it had identical mitochondrial sequences to the stripy one, we can therefore deduce that the mom is this species and the dad is this species, and they both come together to produce this hybrid. So based on this, we looked at every single specimen, every single example of hybrids that we can find, and we found that up to 48% of all angelfishes are capable of hybridizing from all but one genus within the family. So from 7 out of the 8 genera. And this is the highest incidence yet for any coral reef fish, as far as we know. But one caveat with this approach is that it's not suitable for cryptic taxa. So taxa that are not colorful, taxa that are not easily identified via visual analyses, taxa that live in deep coral reef matrices that are very difficult to sample for. So there is still a huge amount we don't know about hybridization on coral reefs, but at the same time, we're learning so much about this supposedly well-known phenomena, at least in the terrestrial sense, that we previously know very little about in the marine context. So that sums up the first part, where we're learning about new things, from ranging from new ecosystems to new species living in said ecosystems to very general processes such as hybridization. So I'll move on to the second part of this talk now, which features some of the systematic work that I've done. And although I've worked on other taxonomic groups, most of the systematic work I've done so far have been focused on the wrasses, in particular wrasses from two main tribes, the Julidae and the Cirrhilabrus. But before that, let's quickly recap some salient biogeography, and this could be very boring. So for those of you watching at home, if you want to tune out now, please go ahead. I don't mind at all. No offense taken, none whatsoever. And because this will feature quite heavily in the next few slides. So I just wanted to get it out there. So we're going to discuss historical biogeography. And to do it in any detail is beyond the scope and time that I have for this talk, or possibly even ever. But one thing that I want to quickly touch upon is the Indo-Australian archipelago, which is also known as the IAA. And this is a region that comprises more than 20,000 islands at the boundary of the Indian and Pacific Oceans. In particular, the Sunda Islands play a major role in the story behind the IAA. And this is one of the most important biogeographical features influencing the distribution and evolution of contemporary fauna in coral reef fishes and also in terrestrial ecosystems. And it's almost single-handedly responsible for the evolution of geminate species, otherwise known as sister taxa, found on either side of the Pacific and Indian Ocean basins. Now, the formation of the IAA goes back to the Paleocene, but with many coral reef taxa, it is events that are centered around the rising and lowering of sea levels during the Pleistocene interglacial periods that is of particular significance. And this will have many flow-on effects that will influence how fishes are distributed and how higher taxa have evolved. And for the Julidae, I focus on the systematics of two species groups from the genus Pseudogeloides. One of my favorite genera of wrasses. Pseudogeloides is a fairly small genus with about maybe 20 species distributed across the Indo-Pacific. They're very slender, so sometimes people call them pencil wrasses. And most of them are tropical coral reef species, except for those in the Pseudogeloides elongatus and the Pseudogeloides ardevi groups. So on the left panel, the three kind of greenish yellow ones, they're the Pseudogeloides elongatus wrasses. And the ones in the right panels with the really dark body, they're the Pseudogeloides ardevi members. And members of these two groups are quite plesiomorphic compared to other Pseudogeloides in that they possess several ancestral characters. And these were the two groups that I focused on. And both cases present fairly standard taxonomic and systematic conundrums that deal with the possibility of cryptic species. And for those who may not be familiar with the concept, cryptic species is a term we use to describe the possibility of two or more distinct species hidden within a single taxon. And oftentimes these may not be very obvious because populations may have very few or nuanced differences in terms of external phenotypes, but they may be separated genetically or biogeographically. So in the case of Pseudogeloides elongatus, it's a widespread species that's found in Japan, Western Australia, and also Eastern Australia, but they're not connected at all. So we refer to this population as disjunct, in that all three populations belonging to one species are not connected. But in this particular situation, it's a little bit more interesting because they also exhibit a secondary biogeographical pattern of distribution known as antitropicality, in that they inhabit temperate reefs in the northern and southern hemisphere, but they're not found within equatorial waters in the equator or in the tropical regions. So the takeaway from here is that we eventually split the single species into three different species. So Pseudogeloides elongatus is restricted to Eastern and Southwestern Australia. And the Western Australian population is elevated to a new species known as Pseudogeloides crux. And the Japanese population is now known as Pseudogeloides paradisius. And antitropicality was a topic that I very briefly dabbled in during my honors year. And to very quickly summarize, it describes a unique pattern of biogeographical distribution where taxa have disjunct distributions on either side of the equator, but not within tropical or equatorial latitudes. So sometimes you'll see the term anti-equatorial being used as well. And that's just a more specific definition of anti-tropical, where taxa are not found within equatorial latitudes, but may be found in tropical latitudes instead. And Bill Ludd, who's like the master of antitropicality, contributed to this study where we looked at the population structure of and molecular ecology of microcanthus. which is a genus of fish that I worked on during my honors year. And the thing with antitropicality is that there are a whole bunch of competing hypotheses explaining this strange pattern, and nearly all of them are in conflict with each other. And for this particular group, we looked at single nucleotide polymorphisms or SNP data to try and reconstruct patterns of dispersal that might explain this biogeography in microcanthers. And we worked out that they most likely traveled north across the equator from southeastern Australia, but with the equatorial populations likely going extinct after the Pleistocene. Unfortunately, the processes that underpin patterns of antitropicality are quite different across taxonomic groups, and it's likely that pseudogeloides, so the wrasses that I shared before, may not share this same historical biogeography. And in fact, it's quite possible that several similar patterns of biogeographical processes across taxonomic groups may not necessarily reflect similar historical processes. I'm not sure how much of that you got, but if you have any questions, please feel free to write down and I'll try and address them as best I can at the end of the talk. Right, so now we're going to talk about the fairy wrasses, which are my favorite genus of fish ever. For those of you who follow me on Twitter, for those of you who know me in person, it's all I ever talk about, fairy wrasses 24/7, all day long. I can't get enough. Yeah, so I'll spend the remainder of my talk today talking about the fairy wrasses, which have been my main interest and a topic that is still an ongoing area of research for me. The fairy wrasses are presently the most speciose genus of Labrids with over 60 species, and they account for nearly 10% of all extant wrasses. They are pan-tropical Indo-Pacific in distribution, so they're found across all the major warm bodies of oceanic bodies except the Atlantic. And they're primarily found between coral reefs between 10 to 250 meters in depth. So not only are they widely distributed geographically, they're also quite widely distributed in their habitat range. And the taxonomy and systematics of this group is constantly being updated with regular new species discoveries and new methods of phylogenetic interpretation. So here's a general biogeographical map of the fairy wrasses, and you'll notice that they have a huge general range, but with a very disproportionate and asymmetrical Indo-Pacific distribution. Now, what this means is that although the range is fairly large, more than 60% of the species are found in the coral triangle, which is that red dotted line that I have in the middle of the map. And as you move peripherally away from this core region, species diversity drops quite a bit. For example, there's only one species in Hawaii and 2 in the Red Sea. And the colors here, I just want you to know that they just correspond to different biogeographical realms, and they're not really of any relevance. But the main takeaway message is that although they have a huge range and there are tons of species, they're not equally distributed across the world. And I've been interested in the fairy wrasses for quite a while now. And actually in 2015, God, this must be so long ago, my friend and I tried to infer the first ever phylogeny. And I'm using very heavy, you can't see my face now because I'm sharing in presenter mode, but I'm using very heavy quotation marks here, using coloration characters and superficial morphological characters to try from our armchairs at home with our Google Science to try and reconstruct the phylogeny of the fairy wrasses. And it was, it was one of those, like I said, armchair nerdy type of science that we do because we love fishes. And in 2018, so 3 years after that, using only mitochondrial DNA, we inferred the first genus level phylogeny of the fairy wrasses. And to our surprise, many of the relationships proposed in our Google armchair phylogeny actually held. But one of the main issues with using a data set like a mitochondrial DNA, in this case, cytochrome oxidase 1, or any type of mitochondrial data really, is something known as haplotype sharing. And this isn't really an issue that is unique to the fairy wrasses by any means, but it is one issue that is fairly common for this group. So haplotype sharing basically is when 2 species share identical barcode regions in a particular gene of interest. So in this case, we're using mitochondrial CA1. And here's the tree, the same tree as before, but in a circular format and with the branches colored by clades. And here's an example where you have 4 morphologically distinct species from non-overlapping regions of the world, but with identical mitochondrial sequences. So for those of you who don't know how to read trees, when we look at genetic distance, we look at horizontal branching, not vertical branching. So the longer the horizontal branch, the more genetic change we have between 2 or more species. So in this case here, we have 4 species and they all look super different and they all live on different sides of the world. They cannot hybridize, they never meet each other. They have never seen each other in their lives, but they have identical mitochondrial haplotypes. And so they exhibit no genetic difference at all in this particular marker. So this phenomenon can be caused by two scenarios, incomplete lineage sorting or introgression. But oftentimes discerning between the two can be quite difficult. And I won't get into too much detail here because it's quite a complicated process. And to make things worse, many morphological characters for the fairy wrasses are quite plastic and quite uninformative. So you can imagine that there's a lot of incentives here in trying to understand the relationships in a group that is so successful, so speciose, yet diagnosed by very few robust characters, and where traditional methods of molecular dating and molecular phylogenetics have proved to be unsuccessful. So this year we published the revised phylogeny of the fairy wrasses, and to overcome the issue of haplotype sharing, we use ultra-conserved element target capture and fortified the data set with mitochondrial CO1, bringing the total taxon sampling to include almost 80%, no, not almost, to include 80% of all extant fairy wrasses, which is the most taxonomically complete data set yet, with all species complexes represented. And for those of you who may not be familiar, ultra-conserved elements are, as their name suggests, very short, highly conserved regions that are interspersed throughout your DNA. So these core regions are very, very conserved, even between related taxa. But as you move away from this flanking region, sequence variability increases. And so it's this combination of conserved core regions and variable flanking regions, as well as the abundance of UCEs in general throughout the genome that make them such a good candidate for resolving relationships of taxa across both shallow and deep timescales. So we took two different approaches with this data set. The first was concatenation of the data, in which all the UCE loci were combined into a single alignment and then analyzed. And we used Bayesian inference and maximum likelihood for this. And the second was a summary coalescent approach, in which every single UCE loci was interpreted as a single species tree, and then an overarching gene tree was inferred from each individual, an overarching species tree was inferred from each underlying gene tree. So again, this is very dense, and I apologize for this, but when I made this talk, I had kind of wanted to use this as my exit seminar for my PhD this year as well. So I modified it a little bit, but there are still some really chunky slides in here, so bear with me here. One surprising result here is that the concatenated data set fared much better than the summary coalescent data set. And what do I mean by fare better? I mean that in nearly all our concatenated data sets, species complexes were always recovered as monophyletic with strong node support. And this was true even for situations where we dropped the number of UCEs to only the strictest filtering. For example, 95% filtering, where we only retained 47 UCEs out of the original 991. So we only retained close to 50 out of 1,000. In contrast, trees inferred using a summary coalescent approach were consistently recovered as polyphyletic or even paraphyletic or even both, with several clades completely unresolved, no matter how stringent our no matter how stringent or relaxed our data filtering. So we decided to dive into the UCE data a little bit to try and figure out why this was happening. And we found that only five UCEs had enough number of parsimony informative sites to resolve our tree with the number of branches that it had. So we found that on average, the bootstrap support for each tree was only about 45 to 55%. So very, very little. And most of these analyses, the sensitivity analyses that we've done so far, such as the GGI analyses and the Robinson-Foles matrix, suggest that the low information content of each UCE locus was responsible for estimation error using a summary coalescent approach. But to be sure this wasn't a result of incomplete lineage sorting, we perform a gene genealogy interrogation. And basically this analysis takes into account all possible topologies that were supported by each UCE gene tree. And if there were incomplete lineage sorting, we'd see some gene trees showing higher statistical support in favor of those topologies over others. So that would be in panel D, but we only see one overarching gene tree that corresponds to the species tree. So in this case, it suggests that gene tree incongruence was caused by estimation error as a result of low information signal. And in such situations, a concatenated approach is favored over a summary coalescent one. Now, I don't know how much of that you got out, but if you're still here with me, thank you very much. I appreciate it. Yeah, and so we also dated the tree and reconstructed the historical biogeography of the fairy wrasses. Now, I'm not going to show you the dated tree because I've already tortured you enough with that really boring explanation of incomplete lineage sorting. But basically, the fairy wrasses emerged in the Miocene, so around 9 million years ago. But their diversification only peaked much more recently during the Pliocene. So there's been a bit of a gap for about 7 years. and with most of the diversification occurring within the coral triangle. So if you remember, that's the dotted line here, where most of the species are found. And if you can remember what I've talked about during the earlier slide explaining the IAA, during the Pliocene, the entire central Indo-Pacific region was characterized by repeated rising and lowering of sea levels, which acted like a species pump as both drivers of dispersal and also barriers of isolation. And in particular, the Indo-Australian archipelago, which serves as the confluence for both the Indian and Pacific Ocean basins, experienced a lot of this sea level fluctuation. So this allowed species to move from one ocean basin to the other. So this blue line here. And once there, the fairy wrasses continue to diversify and disperse, with a smaller number of species reaching as far as the Red Sea, which is the little insular patch of water here, just above the Horn of Africa. And this is actually a fairly common scenario reflected across multiple coral reef groups. So again, like I said, it's not one that's unique to the fairy wrasses. But because of the recent diversification of the fairy wrasses, much of this activity took place within their center of origin, which meant that there's very few opportunities for sustained, more widespread dispersal, which is probably why we see them all over the Indo-Pacific, but with a disproportionate number of species in the peripheral areas, in spite of the large global range. So in addition to providing a broader context to global coral reef evolution, some of the relationships from the systematic study of the fairy wrasses have also allowed us to make more progress in the taxonomy of this group. And one genus that was long suspected to be a member of the fairy wrasses, which was put in a separate genus all on its own, is Cornelia apterygia, which is this species here. And this is one of the few wrasses, I think it's only one of two wrasses, to lack any pelvic fins. So it doesn't have the two little paired fins near its belly. And this is quite rare in the family. And so our studies strongly suggest that actually this unusual taxon is just an atypical fairy wrasse, and that the unusual pelvic morphology is probably just highly derived. So based on that, we did a bunch of CT scans, and you can see that in Cornelia, it doesn't have, that's the middle one, panel B on the right, it doesn't have any pelvic fins, doesn't have any pelvic bones. And we propose just putting it back in with all the other fairy wrasses as one of the species, one of the girls, just a little bit more atypical than everything else. And here's another example of a follow-up study where we implemented taxonomic changes based on results from our UC study. And here we teased apart a bunch of new species that were hiding under a single name, and which we could not do before due to the lack of phylogenomic signal in CO1, mitochondrial DNA alone. So overall, a really nice study with really nice flow-on effects from start to finish, using integrative data sets that combine both genome-wide data, morphological studies, and historical biogeography to help us make informed taxonomic decisions, as well as in informing us about how coral reef fishes in general evolve across the IAA over the Pliocene. So in doing so, basically that's all I wanted to say. And we're almost at the hour mark, and I will save a little bit of that for Q&A, but I just wanted to end off with the Labridae Genome Project, which I'm currently working on with a bunch of really, really smart people at my university, people that are way smarter than me, people that are super talented, Kyle Ewart, Fred Jaya, my principal investigators, Professor Simon Ho and Nathan Lowe. And in this project, we've gotten some funding, and we actually made quite a bit of progress since we started, since I made this slide really, where we have sequenced the genome of one of the fairy wrasses, in this case, Cirrhilabrus aquamarinus, and also a parrotfish. And so we're hoping that these genomes can help us understand a little bit more about the biology and evolution of these wrasses, in particular parrotfishes, which for a long time have been put in their own family. But we now know today that parrotfishes are wrasses. So if anyone ever tells you that a parrotfish is not a wrasse, you can tell them that you've heard it from me personally, that parrotfishes are wrasses with very modified teeth. And so with the genome, we're trying to understand why they have, such remarkable beaks, which they use to scrape at coral and rock. So we have some preliminary results, but it's not ready to be shared yet. So I'll leave it at that. And I just want to thank all the contributing museums that I've worked with over the years: James Cook University, the Australian Museum, Western Australian Museum, Bishop Museum, the California Academy of Sciences, the Australian Research Council, University of Sydney, Lee Kong Chian Natural History Museum, you name it. thank you all of you. And also thank you to everyone who has contributed to this talk and to my research in general. So I will stop sharing here and I will turn my video on, and feel free to ask me any questions if you have any. Thank you very much.
Thanks, Yikai. So feel free to ask any questions. If you would like to ask verbally instead of typing your questions, you can use the raise hand function, and then I can unmute yourself.
Okay, so I can see from the chat that there are tons of questions. Maybe I will start with answering those here first. And then once I've gone through all of them, if there are any other questions, feel free to either put them in the chat or ask me verbally. Okay, so all right, Miguel, is the mesophotic zone the same as the mesopelagic zone, or is it still considered as part of the epipelagic zone? Oh, that's a very good question. So basically the mesophotic zone is anywhere between 30 to 150 meters, but because we're working on reef fishes, we mostly look at fishes that live very close to the substrate. So yeah, it could, if it's within 30 to 150 meters in open water, I guess you could call it mesopelagic, but I've never heard that term used before. This might be a question better suited to Luis Rocha. I can ask him this for you and I can get back to you if that's what you want. But yes, from my understanding, it's as long as it's within 50 to 150 meters, it's a mesophotic. Actually, some scientists and Carol Baldwin's group, who's also very good at working on these deep reef organisms, they classify the lower mesophotic as a different term known as the raryphotic zone. So there's a lot of papers out there and they're very easy to read actually on mesophotic, on raryphotic reefs. So they might, be of use if you want to know more about them. Hope that answers your questions. James, hello, James. At what point do hybrids become distinct species rather than simply a distinct hybrid? Is it a function of being more genetically, morphologically distinct from the two species that produce the hybrids? And then Miguel's response to James is probably when their populations become distinctly isolated from the parental lineage, which would lead to speciation. So yes, Miguel's response to James is actually quite correct. So James, when two species hybridize, they will produce an offspring, and that offspring may or may not be fertile based on how much genetic separation the contributing parents have. So a general rule of thumb, and this might not be general at all, but from what we know is that if the parents are more genetically different, there's a higher chance that the hybrid may not be fertile. So in a lot of hybrids that are able to breed with other hybrids and also with their parents, it's usually between two parental species that are very closely related, or in some instances completely introgressed. So in these situations, if the hybrids that are formed are able to, mate with other hybrids and also other parents, if they, for whatever reason, become isolated long enough, they might eventually speciate into something else. But in the context of coral reefs, there are very few proven and testable examples. It's something that I've been interested in looking at because in Guam is a very nice natural example where we potentially have a few species there that are hybrid derived. But anyway, I don't know much about that because I haven't really looked at it yet. But yes, if they are fertile and if they are isolated long enough, they may eventually speciate. Hello, Kang Ming. Are the hybrids fertile and do the hybrids introgress? So yes, it depends. Not all hybrids are fertile. Some of them are. If the parents that are forming the hybrids are very, very closely related, then there is a possibility that the hybrids are also fertile. And in that case, they can either mate with other hybrids to form more hybrids, or they can mate with either parents to form paternal back crosses. And in certain areas of the world where hybrids are more common than the parents, this can be quite detrimental, and it can cause local extinctions because hybrids that are fertile can keep repeatedly mating with parents, and over time, pure species may be bred out entirely because they just keep getting introgressed with hybrid DNA. So yes, that is a possibility. Another question from Miguel: do the UCEs mean that the two or more species have a common ancestor node in the phylogenetic tree? Yes. So the core region of the UCE is very conserved, especially between sister taxa. And the further two species are in terms of their relationship, the more variability you see in the flanking regions. So if two species are very distantly related, for example, a wrasse and a damselfish, then using UCEs might not be a good thing because there's just too much difference and it's not possible to reconstruct a phylogenetic tree. Stephen, good job with describing the science of genetics and biogeography to keep everyone on the same page. Well done. Thank you very much, Stephen. Another question by Miguel. During the Miocene period, were there areas that were isolated leading to allopatриc speciation? The Miocene is, yes, a lot, actually, yes. So there is a paper by Peter Kalman and Dave Bellwood on Miocene level, exploring evolution of wrasses, butterfly fishes, and damselfishes during the Miocene. And yes, there was a major period where a lot of reef fishes started diversifying based on allopatry. And then the Pliocene kind of is a more secondary area where, you know, speciation was more prevalent at the tip level. So the Miocene was categorized, if I'm not wrong, by deeper levels speciation events, and then followed by the Pliocene. Okay, there's a lot of questions. I'm going through them as fast as I can. Another question by James: Is genetics the main proof that taxonomists use to determine if species is not a distinct species? You've mentioned comparative morphology and historical biogeography, but is it mainly genetics? And at the end of the day, who determines if something is a distinct species? Is it official once a paper describing a species is published? Okay, so as a taxonomist, I personally like to use multiple lines of evidence. So genetics is not the only, is not the end all and be all. And as you can see with fairy wrasses, if you were just to go by genetics alone, there would be a lot less than 60 species of fairy wrasses because so many fairy wrasses have very similar or completely identical mitochondrial haplotypes. So we use a combination of things, and basically it's just the more lines of evidence you have separating and justifying why two species should be different, the better. And that could be anything down from coloration characters, morphological characters, biogeographical patterns of distribution, genetics. So it's up to you as a taxonomist making an informed taxonomic choice, whether or not two species is or is not different. And as to whether or not there is an official body governing whether something is new or not, the answer is no, and the answer is other taxonomists. So you and I can describe a new species today, which we believe is new, and then, Dr. Gill or Dr. Tan may not agree with it, and, they might just, in a follow-up paper say that, oh, we don't believe that this is a new species. So it's up to it's up to other taxonomists whether or not they want to perpetuate your findings. So if something is valid, and people keep using it over the years in reviews and in books and in papers, then it stays. But as you know, taxonomy is always in a state of flux, and people can sink species and people can resurrect species. So there is no official body, I guess, at the end of the day. A question by Yusri: Are those hybrids common in the wild? I only saw one hybrid of butterfly fish during my whole dives from 1998 in Malaysia. Yeah, they can, or they may or may not be common depending on where you go. Like I said, if you're diving in an area like a known hybrid hotspot, for example, between two distinct biogeographical realms, then hybrids may be more common at times than their parental species. If you're diving in Christmas Islands, in Cocos Island, for example, almost every fish you see from the genus Centropyge is a hybrid. They're very common there, but in other places, they're not so common. And in angelfishes, hybrids are really, really rare. So when we did our angelfish study, we didn't talk anything about the abundance of hybrids. We just talked about the occurrence of hybrids. Question from Abel: My son just told me that parrotfishes look like wrasses last week. Now I can tell them they are. Yes, you tell your son and everyone else that parrotfishes are wrasses. This is this used to be a very contentious thing, but people are now warming up to the idea that parrotfishes are wrasses. Another question by Miguel: You mentioned that 7 out of 8 species of wrasses can be hybridized. Why is that one of the hybrid wrasses not? Why is it that one of the wrasses species cannot be hybridized? Oh, so sorry, I might have misspoken here. I mentioned that 7 out of 8 genera of angelfishes have been reported to hybridize. So there are 8 genera of angelfishes. Only 1 have not been reported to form hybrids, and that is from the genus Pygopleides. And there's only 1 species in Pygopleides. It's a monotypic genus, Pygopleides diacanthus, and so it can't hybridize. So it can't form intergeneric hybrid. So in order for Pygopleides to hybridize, it would have to hybridize with another genus of angelfish, which is very, very rare in marine fishes. So that's why it's the 1 out of 8 that has not been documented to form hybrids. Thank you, Kai. Interesting that parrotfishes are derived from Labridae. That means that parrotfishes, wrasses, evolved earlier than parrots. That's a really good question. So parrotfishes are derived labrids, but they did not derive from labrids. So when taxonomists and systematists talk about something being derived, they're just saying that they have characters that are not the ancestral state. So in wrasses, having teeth that are not fused together is the ancestral state, which means that all wrasses, well, most wrasses, that's the default setting, that wrasses have teeth that are kind of like caniform in their morphology and not fused, but parrotfishes have teeth that are fused into a beak. So this character is secondarily acquired, so it's a derived character. So they are still wrasses and they're nested within the wrasse tree of life, but they didn't come from wrasses. Yeah, hopefully that clarifies. David, it's difficult for me to think of a natural situation in which hybrid offspring gets completely separated from the parents though. Do you know of any examples where this happened? That's a really, really good question. And yes, it is very difficult. It is possible if hybrids at a specific area are so common and fertile that they are able to hybridize with other hybrids and produce fertile second generation hybrid offspring. And if these larvae and eggs were to, for some whatever reason, float and wave into a separate island, far enough away from the main colony, but not far enough for them to reach, and then establish there, and then form a big enough colony that they're able to self-sustain themselves, then that could be a possible speciation event. Like I said before, it's super rare, and I don't know of any examples of this happening in the wild, but Guam is a potentially good area to investigate this, because the angelfish, the endemic angelfish in Guam, Centropyge shepherdi, is very similar. It's almost uncannily similar to a hybrid between Centropyge loricola and ferrugata found in the Philippines. So in the Philippines, 2 angelfishes hybridize occasionally to form an offspring that look almost identical to the species known as shepherdi in Guam. So there have been a few talks about whether or not shepherdi is a hybrid derived species from one of the Philippines, hybrid gene pool or whatever. We don't really know. We're speculating at the moment. We don't have enough time or material to test it. But yeah, it's rare. Thank you. I like the Rafflesia behind. Thank you. I love this very much. This is my favorite thing in this entire room. I'm actually zooming from the Lee Kuan Yew Natural History Museum, and there are a whole bunch of really nice murals, including some butterfly ones, but I chose this one because it's very striking and I like Rafflesia. Well, there are a lot of questions. I'm not sure if I can answer them all. It's already 9. Okay, another one by Abel. Since many reef fishes breed by broadcast spawning, is it surprising that hybrids are relatively uncommon? Yes, which is why we started the hybrid paper, because there are so many fishes living in overlapping distributions, and because there's so much broadcast spawning, why haven't there been enough hybrids reported from fishes that live in such areas? And so the angelfish paper kind of, pun intended, spawned from that idea. Jane, thank you very much for the brilliant talk. Best wishes from England. Thank you, Jane. Glad to see that you're here. Philip, do you expect us to continue to see newer hybrids of wrasses with the way our oceans continue to warm? Possibly. In Australia, for example, I live in Sydney, and it's right at the boundary of the temperate zone. And every year during summer, we have species that flow in from the Great Barrier Reef as larvae, and they grow up as juveniles in Sydney, but then they die off during winter when it gets too cold. But in the last 10 years or so, we're seeing a lot of these juveniles actually grow up into little baby fishes, and they're lasting the entire winter because it's getting warmer here. So we don't know what's going to happen, but we have actually seen 2 species of damselfishes from Queensland and Sydney hybridizing in Sydney that were not meant to be here. So possible, we don't really know. Last question, have we finally put Premnus bioculatus in the amphiprion? Yes, James, it's the spine chicken anemone fish is now in amphiprion. Catalina's direct message to me, do I eat fish? Yes, I do. I eat fish. It's one of my favorite foods. And the rest of the talk, rest questions are just thank you very much. Thank you guys. Okay, I really appreciate you coming here. I really appreciate you spending the time with me on this Thursday evening. Thank you for the questions. If you have any other questions that I haven't answered to your satisfaction or at all, you can find me on Twitter at Kai the Fish Guy. Yes, so yeah, that's all from me. Thank you very much.
Thanks, Ikai, and thanks everyone for the flurry of questions. If you have any questions, I guess, you can contact Ikai at Twitter. Maybe you can leave your, you can type in your Twitter handle in the chat box.
Yeah, so my Twitter handle is at Kai the Fish Guy. I also have an Instagram with a similar handle. Yeah, thank you very much, Jia Xuan. So feel free to message me there. Feel free to email me. I think my email is on my Sydney University profile, which you can find very easily. Yeah, I don't have anything else. Thank you very much for coming.
Okay, thanks everyone. Have a great evening or night.
Miguel, are you using eDNA techniques to identify fishes? One of my colleagues and my boss at the Australian Museum, Joseph Di Battista, he's one of the foremost authorities on using eDNA techniques. And they do use it to sample fishes, but eDNA is kind of more of a broad detection device where they identify what kind of fish is living in your area rather than new species of fishes, because it's down to using barcoding. And yeah, it's more of a general broad application science as far as I know. I don't think anyone has been using eDNA to identify new species. I think the only time a new species has been identified using eDNA is when they discovered that new ruby pipe seahorse pipefish, the ruby pipefish from Western Australia. Yeah, but I need to read up more about that because I'm not very familiar with seahorses.
Thanks so much.
No worries. Okay, I think it's 9:06, so I'm going to leave you guys. Again, thank you very much. And I'll see you on Twitter if you have any questions. Otherwise, have a great weekend. And yeah, thank you. Bye-bye.
Okay, thanks everyone.